this is Miss Litton on election day. Yay! It will soon be over. And we are, this is Honors Bio, and we are reviewing Chapter 7, Cellular Respiration. So I'm going to quickly go over the big ticket items, and we'll dial it down to each of the key concepts to make sure you got it, right? So on the big ticket items, we are doing what to glucose, oxidizing or reducing? We're oxidizing the glucose, and we're temporarily giving the electrons or energy to who? Yeah, NAD and fat. NAD and FAD, who will take them to the top of the ETC, so we can right? Reductase, cytochrome oxidase, concentrate our hydrogen ions so they can go back in, and make some ATP. Okay. So glycolysis is the first step, whether you have oxygen or you don't have oxygen, and it always occurs in the cytoplasm. Good. And it has 10 different steps, which means it must have 10 different enzymes. Good. And you end up with something called pyruvate. Now, pyruvate has choices depending on if there is oxygen or not. Okay, the next destination, the step 2. If you have oxygen, you're going to go into transition prep, and you're transitioning into the mitochondria, and that is really the most efficient way, and that gets you the most ATP, potentially 36 to 38, but probably efficiency-wise it's less than that, 32 maybe. Okay, but that maximizes your ATP potential. If you have no oxygen, or you don't have enough oxygen for all your pyruvate that you generate, then you're going to transition into fermentation. And how you ferment, that second step, you and I, we'll make lactate or lactic acid, which we associate with our muscles being what? Sore. Other organisms, plants and yeast, they'll take the pyruvic acid and convert it to alcohol and carbon dioxide. Okay? So we just want to run through the steps and make sure we understand all of them. So we're going to start out with glycolysis, and we'll go through and do the songs and everything, make sure you understand it. But let's remember, the song is there to support and to be a scaffolding to our understanding of the chemistry. So the key events in glycolysis is breaking glucose in half. You make 2 NADs and you make 2 ATPs. Remember, these ATPs are not a oxidative phosphorylation ATP formation. This is substrate level phosphorylation, a direct handoff, right? No electron transport chain involved here. Okay. Now, do we only make 2 ATPs? What do we really make? 4. But because we had to spend how many? 2 to get the party started, we just say, oh, we made 2. Okay? Now, these 2 pyruvic acids still have energy in them. Okay? And if possible, we will go on and we'll do transition prep, which will bring us into the mitochondria. And now we want to review the idea of what the different areas are of the mitochondria. And so there are 2 areas, 2 areas. Okay, we have this intermembrane space, and what are we going to concentrate in that intermembrane space? Hydrogen ions, okay? And where are those hydrogen ions ultimately coming from that we're concentrating right here? Where are they ultimately coming from? Glucose. Okay, and you were saying NAD and FAD, right? But NAD and FAD got them from glucose. So these hydrogen ions you see right here ultimately came from glucose. Okay, it is a double membrane structure, and the red line I'm generating right now, this is where all the ETCs take place. And so you concentrate these hydrogen ions to the inner membrane space. Then when they go back in, that's when you make your ATP. Okay? The interior portion inside, this region all inside of here, where the Krebs cycle takes place, where the transition prep, this is called a matrix. Okay? So those are the 2 regions. Glycolysis is occurring, though, out in the what? Cytoplasm. Transition prep is occurring inside the matrix. The Krebs cycle is occurring inside the matrix. How many times are we going to do the Krebs cycle? Twice. Good. And the electron transport chain occurs on this red line right here. Okay, there is a line out here. That's just the outer membrane of the mitochondria. Any questions on location and events? Yes.
So ETC happens on that red line.
Yes.
What is that red line?
That's the inner membrane of the mitochondria, the most inner membrane. And the space between the outer and the inner membrane, that's the area I've colored in green, and that's the region where the hydrogen ions are concentrated. Who concentrates them? Reductase, reductase, cytochrome, oxidase. So when they come back in through ATP synthase complex, that's when you generate ATP. Strong, powerful NADs are generally, say you get 3. It's less than that, but about 3 ATPs. Weak NADs and FADs only get you 2 ATP when you go take them to the bank. Yes?
What about the cristae?
Okay, the cristae, good, I'm so glad you said that. The cristae are the folds on these folds on the red line. Yeah.
Okay.
Yeah, they're the folds on that red line. That's good. I'm glad you brought that up. Okay, any other questions about location and reactions? Okay, so let's answer these questions you've seen, and then I have some that you haven't seen. So try these out. My SpongeBobby and SquarePantsian. Some polls have closed, have they not? Do we have any early election results?
Maybe East Coast.
Yeah, isn't it? Well, East Coast and Central. They want that right there.
The first poll's closed
At
at 3.
3? Oh, okay. I thought we were getting close. Okay. Ah. Okay, Krusty Krab Pizza. Let's go.
I submitted one.
Oh. Good job, Invisible Boatmobile Campfire Song. I can't do anything right because it's Higgles. All right, let's see what people said. Let's help them. Oh, some people forgot about C. Remember? Remember, who does it? All cells, where? In the cytoplasm. Okay. Now, let's do better than we did the first time on this question, eh? Remember another word for needed? What could we be writing instead of needed? We could be writing reactants. Do you have an ed puzzle you need to do tonight? Some of you already do it. I totally chopped that ed puzzle too. Made it very short.
Oh yeah, the last question.
It was supposed to be like 14 and I made it 7.
Thank you.
It was going into the details and I thought, you know what, I'll just handle it. So I was like, he's talking, I'm all done. Crap. Will we do better? Yes, we will do better. I'm so proud of you. Good job, holographic. Bring it around town, I'm ready. Let's get another hundy. Yeah, she swam on over. Just say, huh? She got lost. She asked somebody for directions. Went the wrong way. Will we get another 100? Will we? No. Some people forgot G and some people forgot H. Okay, so yes. ATP is a product. How much ATP do we make? 4, but we sent out 2. Good. And then what else did some people forget? H. Pyruvic acid. Yeah, definitely pyruvic acid. All right. Preparatory reaction. If you remember the chemistry behind this, we wanted to understand that pyruvic acids each have 3 carbons in them. And we could follow those 3 carbons. When we go in our song, we're pulling a carbon off each of those pyruvic acids, but when we're pulling it off with a couple of oxygens, so it's carbon dioxide. Okay, so here are your 1, 2, 3 carbons. And where that 1 carbon is, now we're putting CoA in its spot, and that CoA is going to help feed it into the Krebs cycle. Now, if I follow the CO2, which would be here and here, right? What's the one thing that's missing? The hydrogen, and where did it go? NADH. So again, pulling energy out of ultimately the glucose and giving it to NAD. Remember, NAD, I can't take that to Taco Bell either, okay? But I can cash it in for some dollars, right? It's still a check, just a smaller check, right? I can cash that in at the ETC for some ATP. Did I make any ATP here? Do you see any ATP here? No, because I didn't make any. Okay. Did I make ATP in glycolysis? Yes. Have I used oxygen yet as a reactant? No. I don't need oxygen here. I don't need it in glycolysis. I don't need it till the bottom of the what? ETC. I haven't even used any oxygen yet. Okay, I don't want you to memorize this and stress out about it. I wanted you to understand it. Okay? So don't sit there and go home and memorize. You're wasting your time. What you want to get is the big picture. You're removing a carbon. We're doing the bit, right? Basically doing the bit. We're reducing an NAD and pulling off a CO2, and we're decreasing the carbon count by each one by 1, right? Bye, sweetheart. And we are attaching, in this case, what makes it different is we're attaching a CoA in that spot right there so we can go in and do the Krebs cycle. All right, now, when we go to the Krebs cycle, okay, and I'm going to just jump to mine, okay? These right here, this, I don't know why I drew it like that. I was thinking about underlining that. Okay, this C2 right here, that is this C2 right here. This is what's going on to the Krebs cycle, that C2. You follow? Okay. So already preloaded as a C4, we hook that together. Are there hydrogens and oxygens on these guys? Yes. I'm just not showing them to you. I'm just simplifying it down, okay? The 2 and the 4 get together. That makes sense. It makes a 6. What is this right here? What are we calling that? We're calling that do the bit, right? And in our mind, we know we're removing a CO2, reducing an NAD, writing another check here, right? And then we do it again, write another check, and we're down to 4. We still can extract more energy out of it because we make an ATP, we reduce an FAD, and we reduce an NAD. How many times are we going to do this cycle? Twice, okay? What's another name for it? Citric acid cycle, good. Now, this is the last step before we go to the ETC. So evaluating everything we've done so far, glycolysis, transition prep, and the Krebs cycle, which one makes the most NAD for us? Krebs cycle. Because how many do we get with the full, when we do both turns? How many NADs do we get? 6. And we get 2 FADs. You could do the math right there. If you get 6 NADs, and how many ATP did I say you get from that? From each? 3. 3 times 6 is 18 ATP when I go to the electron transport chain, right? And those 2 FADs, 1 a turn that I get, generally speaking, those will get you how many ATP? They don't go to the top. Remember, they go in part way down. How many ATP will they get you? 2. 2 times 2 is what? 4. So 18, 19, 22 ATPs just from that, plus I go around twice, there's 2 more, right? That's 24 ATP just from here that I ultimately get. How many did I tell you you would get? About 36 to 38, right? So you're getting 24 of them just from the Krebs cycle. Is the Krebs cycle important? Yeah. Transition, is it going to get you some ATP? Sure. You know, glycolysis? Sure. But this is the cash cow right here, Krebs cycle. So you want to keep that turning. Yes?
So the NAD making 3 ATP and the FAD making 2, that's the part that happens twice?
The Krebs cycle happens twice.
So when it says NAD times 3, that's
Oh,
what
this
happens twice.
this would be at the bank of the ETC. When I go to the electron transport chain, let me jump there. Okay. When you go to the electron transport chain, if NAD comes in at the top, he does reductase, cytochrome oxidase. So he puts out 1, 2, 3 hydrogen ions. So that means in the ATP synthase complex, 1, 2, 3 ATP as a result of it. A lot of it. Or FAD comes in, he comes in a little lower. So he only gets a reductase and a cytochrome oxidase. So he only makes how many?
2.
Or a lot of it. So my point that is that the more NADs you get over to the electron transport chain, the more what are you going to make? ATP. Yes.
So for every side, like for every hydrogen ion on the outside, you get 3 more coming through the ATP synthase.
Yeah. If I crank 3 out here,
Okay.
then that means 3 will want to come back in.
Okay.
If I crank 2 out there, 2 are going to want to come back in.
Okay.
Does that make sense?
Yeah.
Okay. All right. So then when we, let's see. Let's do, let me hit this too. This, I don't know if this will help you. Maybe it will. Take a look at this picture. Okay. Here's glucose to pyruvate, and we directly make 2 ATP, yes? But we also make 2 what? NAD, which when we take that ultimately to the ETC, those 2 NADs are weak because they're made way out in the what? Cytoplasm. So they usually only get us, these 2, they usually get us around 2. So 2 times 2 is what? 4. Okay, these, here we're going from pyruvate to acetyl-CoA. We get 2 more NADs. These are powerful ones, so these are times 3. 2 times 3 is 6. Okay, now we do the Krebs citric acid cycle 2 times. So do the bit, reduce an NAD, do it again, do the bit, reduce an NAD. That's 6 NADs. Each one of those are powerful. 6 times 3 is how much? 18. Every time we do it, we get 1 FAD, so we do it twice. Those are weak, so it's times 2. 2 times 2 is 4. So which part helped the most for getting us the most ATP? That would be the Krebs citric acid cycle is really what we really want to do. Now, to make this work, who catches the electrons here at the bottom? Oxygen forming water. If there's no oxygen, then you can't no, then you can't do Krebs. Can't do Krebs, can't do transition, so all you can do is glycolysis, and if you have no oxygen, then you're going to go one of the 2 fermentation pathways. You're going to either make lactate, makes our muscles sore, or you're going to make alcohol, depending on what kind of organism you are. So the only ATP you get is from glycolysis. So if we look at anaerobic respiration, remember, this is where you take glucose, and running along the bottom here is glycolysis. This is glycolysis along the bottom. You break it in half, costs you 2 ATP, you get 4 ATP out of it, and you reduce some NAD. If you don't reduce NAD, you can't make any ATP. So if all we do is this, and this, and this, and this, all our NAD will be what? Reduced, right? We need it to be back to what state? Oxidized, so we can reduce it again, right? So that's why you do fermentation. The process of fermentation takes your lactate and it reduces it takes your pyruvic acid and reduces it into lactate, or takes your pyruvic acid and reduces it into alcohol, so you can oxidize your NAD and reuse it again. Okay, what part makes you scary? Yes?
What's the ATP synthase complex?
ATP synthase complex is that thing right here that allows your hydrogen ions to come back in.
So it's putting it back into the matrix.
Yes,
Okay.
back into the matrix. We shoved it in the intermembrane space. It wants to be back in. What else? Yes.
So it's going through a protein, or what's happening? Or no, wait, that's a membrane, so it's not.
Yeah, these are, remember how proteins can act to move molecules from one side of the membrane to another? right? We have channels and carriers, right?
But that's what those are. No,
That's
it's
what
not
these
the
reductase,
membrane.
reductase, and
Oh,
cytochrome
okay.
oxidase, that's exactly what they are. Okay, what else? Yes?
So does ETC kind of combine with the Krebs cycle?
It doesn't, the Krebs cycle is taking place here, you know, in your matrix area. It's taking place here, but then when they, in the process of doing the Krebs cycle, you generate a lot of NAD. Those NAD then are right there next to that membrane, and they start that process going.
So alone with ETC, how much ATP is made?
With the maximum you can make is maybe 36 to 38. The efficiency isn't that great. It's
Okay.
usually maybe 31, 32. But ideally what you would say is each powerful NAD gets you 3.
Mm-hmm.
The only NADs that aren't powerful are the ones from glycolysis because they happen to way out in the cytoplasm. And each FAD is generally weak, usually only gets 2.
Okay, so it just takes it to the next step.
Yeah.
Okay.
What else?
How do you get ATP from reducing the NAD?
Okay, so the NAD has the hydrogen, and so it's reduced, and whenever we have the hydrogen, it tends to give you its what? Electron. So what ends up happening is, if I have these 2 electrons, I'm at the top of the electron transport chain, and I'm NAD. I just dropped off the electrons, now I'm oxidized, and I can be used in an earlier step. Okay, now, passes it, electron transport chain, so electrons are getting transported. Now, because he just got this, his name is reductase. He's like, I'm so powerful, and he cranks the hydrogen out to the inner membrane space.
Okay.
Gets pumped along, here's another reductase. Okay, cranks it out. And here is cytochrome oxidase, and cranks another hydrogen out. There you go, it's not very passes it on to the final electron acceptor, who's going to hand these electrons to who? Oxygen forming water. Because reductase, cytochrome oxidase, concentrated these hydrogen ions out there, they want to go back, remember like Disneyland before it opens, they want to get in, so they go in through ATP synthase complex, who snaps a phosphate on to make ATP.
So it doesn't do anything directly, it just does it by...
It's coupled.
Oh, okay, that makes sense.
Right? Okay, more on that. And I have a whole membrane analogy up on YouTube. For ATP, I did like electron transport chain, reduction, oxygen. It's not under cellular respiration because it's also used in photosynthesis. So I just explained that idea. Yes?
What's the difference between reductase and cytochrome?
Just names. That's all you need to know.
What's that coupled reaction again?
The electron transport chain with the chemiosmotic synthesis of ATP.
Okay.
Ask me again. Okay, one time through this, here, let's do this. Okay, when we talk about calories, who has more calories? Fats or carbs? Fats do, and the reason is, carbs are the monomers, sugars, right? And we know how many carbons we can get out of that. How many carbons can we get out of a sugar? 6. But when we have a fat is made out of glycerol and long fatty acid chains. Those fatty acid chains can be 18 carbons long. Snip those into little acetyl-CoAs to put through the Krebs cycle. 18 snipped into 2 carbon units is going to be 9, and you got 3 chains of those, right? Remember saturated and unsaturated fats? So 3 times 9 is what? 27 acetyl-CoAs. Plus the glycerol can go through 2, that's 28. Okay, glucose gets us 2 acetyl-CoAs. I can get 28 acetyl-CoAs from a fat. That's why fat has more calories. It has more energy in its bonds than glucose. So if I eat a lot of fat, I will meet my energy needs quicker, so the leftover gets what? Stored. Whereas if I eat some complex carbohydrates, I can get the energy I need plus fibers that help with your digestive system and all of those things that go along with it that you've read about. Okay? So that's where that comes from. But fats taste really, really good. Okay. So let's start with this, and then I have some other questions to ask you. Oh, I already asked you that, so I'm not asking you that. Let me ask you this. polls closed. It's 3 open.
Where did they close?
East Coast. Well, as long as you're in line, you could still be in, right?
Yeah. This just seems unfair. Yeah, why would you call it 250?
Well, for them it's 6.
Oh, it's 6.
Yeah, so some polls are closed then, not all polls, but some are. Okay, all right, let's talk about it. Okay, good job, Dory, bringing around town, Mr. Sun. All right, so the question is, the greatest contributor of electrons to the electron transport chain is, and most of you got it, the citric acid cycle. And you would say that because you would tell me the citric acid cycle will bring 6 NAD reduced and 2 FAD reduced. You follow? Yeah? Okay, and some people said A, oxygen. No, it doesn't contribute any electrons. It does what? It catches them, takes them away. So definitely not oxygen. Then some people said D, the prep reaction. Prep reaction only makes 2 NAD reduced. So that's not very many. That's going to translate, 2 NAD reduced will give us how much ATP? 6 ATP. Okay, these will get us how many? 18. And this will get us how many? 4. So who did more for us? Citric acid cycle. Questions? Okay. Yeah. It should be a handy because we have talked about it before. Yes.
NAD reduced is the same as NADH, right?
Yes. Sometimes it's written like this. Sometimes it's written like this. Usually like this. Or it's written like this. All the same thing. And the opposite of that would be this. Or just like this. Campfire song. Let's go. Okay, that was scary. Remember this math we've been doing back here? Okay. Each strong NAD gets us how many ATP? 3. Okay, 3. Okay. Reductase, cytochrome oxidase. Hydrogen out, hydrogen out, hydrogen out means hydrogen in. Every time one comes in, you make an ATP. Okay, so definitely not 1. And then somebody said 36. No. Per NAD. Not per every NAD ever made. Just per 1 NAD. Check with your bio buddy. Bell of shame, gong of victory. Do you feel like you have better ownership of the material? Where could you get more practice questions? The books, online. Yes, you could. Who catches the electrons at the bottom? Oxygen forming water.
First place. Let's go. First place.
Good job bringing around town, the invisible Beltmobile and Dory.
Yeah, you were like 16. I was like 19. I had the same goal.
So for like the transition.
That's good. That's good.
Yeah.
Oxygen. Good. That was beautiful.
It is beautiful.
Can you take a picture of that? It's like it's so much.
Two of you got it right.
Oh.
Bring it around town.
Bring it around town. How many NAD molecules are produced during the complete breakdown of one molecule? You get 2 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 NADs. Do you understand now?
Yeah.
Look, a bunch of you said 6. So you only included the Krebs cycle. You forgot the 2 NADs you made in transition prep and the 2 NADs you made in glycolysis. Right? That was a good tell. That was a good question to ask you. You feeling okay now?
No.
Reduce some NAD, make some ATP. There's 2 NADs you got. And then transition prep, and you reduce some NAD. There's 2 more. Then 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an NAD, and you do that twice, that's 6. Plus 2 is 10. Right? Question?
Wait, so the Krebs cycle makes 6 NAD total?
Yes.
Not each time. Okay.
Yeah. Each time it makes 3.
Yeah, okay.
So remember this right here that I was showing you? How many NADs are made through the complete breakdown? 2 from glycolysis, 2 from transition prep, and 2 from the citric acid cycle. 10. 10 total NADs. Which of these is not true? So that's saying all of them are true except 1. All of these are true except 1. Oh.
Want to give us a hint?
Okay, so for fermentation, you're doing this, which is glycolysis, right? And then you're doing this. You're either doing, if you're a plant or yeast, or you're an animal. Okay, one or the other. And here you're either making alcohol and CO2, or for us, we're making lactic acid. Either way, you oxidize your NAD that got reduced here. Now, you only make 2 ATP, and that's happening during glycolysis. Whoa. Hi, keyboard. I didn't know you could make it smaller like that. So, is there a net gain of only 2 ATP? Yes, that's that part's true. Does it occur in the cytoplasm? Yes. Does NADH donate its electrons to an electron transport chain? Do you see any na-na-na-nas going on here? No. Does it begin with glucose? Yes. Is it carried on by yeast? Yes. I think there's one more if you want it. products of fermentation. You're welcome, sweetheart. Have a good evening. Remember, if you're staying up late watching the election and studying, eat some toast. You don't get it? Because I always say that. Eat toast.
Ben's not looking good.
Is lactic acid a product of fermentation? Yes. Makes our muscles sore. Is glucose? No. It was a reactant. Is alcohol? Yes. Is carbon dioxide? Yes. That's done together by plants and yeast. Is oxygen? No. Is acetyl-CoA? No. That's from transition.
It's not true.
Okay, that's it.
I keep
Yay.
hand on my head.
Oh, yeah.
You know, will I? You know what I can do?
You're welcome. I was checking that out. How did you do that? Here, let me stop this real quick. Oh my gosh. It stopped at 1 minute and 21 seconds.
The review of the video.
No.
And it was such a good review.
That reminds me of my life.
Oh my, no.
I don't know why it stopped.
I needed that.
Yeah, we have good. We have great
Our
news.
nose are so good.